The origin of replication, represented by this red line, is a single nucleotide sequence that initiates helicase, represented by the green dot, to use energy to begin separating hydrogen bonds from nitrogenous base pairs in order to unzip the parent DNA strands. This creates a fork or split between the two parent strands. Here, the dark green dots represent single-stranded binding proteins. These proteins line both single strands of DNA in order to prevent the strands from winding and recreating a double helix. In orange, we have gyrase, which is a topoisomerase that works ahead of the replication fork and relieves tension by preventing supercoiling. Here we have RNA primers. RNA primers notify RNA primase where to be placed on the parent strands. A 3' end is needed in order to create a starting point for DNA polymerase. DNA polymerase 3 is depicted by the peach dot. This is an enzyme that adds nucleotides to the progressing DNA chain. This is required for DNA synthesis. DNA polymerase 3 is only able to add nucleotides in the 5' to 3' direction. The purple line represents the leading strand. The leading strand is a continuous addition of nucleotides to the parental 3' to 5' strand. However, the complementary parent strand is replicated with a lagging strand that is discontinuous in the 3' to 5' direction. This discontinuation occurs in fragments known as Okazaki fragments that are a result of the lagging strand waiting for more of the DNA to unwind, since the parental fork only unwinds in one direction. RNA primers are placed in the fragment opening between the discontinuations on the lagging strand. Here, the DNA polymerase 1 is placed to remove and replace RNA with DNA nucleotides. These pockets are then bonded together by ligase. Ligase seals the DNA fragments together by using phosphodiester bonds to create a continuous DNA strand.